Okay, Marinka, Yuri, thank you very much for the invitation. I'm really honored to give you a talk about our research, what we are doing at Bayer. So I will talk a little bit about proteochemometric modeling for COVID-19 drug repositioning. But I cannot start it with giving a small slide about what we are doing at machine learning research at Bayer. So when we started this department, our main idea was always like to build up a group that develop machine learning algorithms or intelligent systems that allows us to transfer information that can be obtained on a large scale from high throughput data into an output or an insight that is typically very expensive. So, for example, if you have a data point on the right-hand side where you know what, for example, what is mode of action of a certain protein is or the toxicity, this endpoint can be easily cost a few €100. So it's very, very expensive, this information to acquire. So therefore, we started very early in developing machine learning algorithms, mostly for the purpose of developing representations. So we are working in actually 4 domains. So we are working in the chemical space, so coming up with unsupervised or self-supervised learned representations for chemicals. We are working in the omics space, but we of course develop also the information that's coming from the transcriptomic experiment and incorporated in drug de novo design. We're working on high content imaging screens where we want to build representations that automatically can annotate and also analyze the very, very large high throughput screens. And also we are working on developing representations for biologicals. So let me briefly walk you through the bread and butter of a computational chemist. So in many cases, the computational chemist is interested to gain insight about the bioactivity of a certain compound, and that is typically called QSAR, that's quantitative structure activity relations modeling. So if you know on the left-hand side what a compound is doing, if it's active or not inactive, then you can have a very, very good idea to maybe boil down this very large chemical space into a smaller part and can use it to prioritize compounds in a hit discovery business. So all this works exploit always the same principle. So that means if you have 2 compounds and they are very close together in the chemical space, so they share a high similarity, then it's very likely that the second compound is maybe also doing the same as the first one. So therefore, all these QSAR processes always work to start with the same workflow. So you collect the data set of molecules and bioactivities that are of interest. Then you always start to extract some computer interpretable features from the ligands for the chemicals. Then you can train whatever kind of machine learning algorithm to predict if a molecule binds to a target or not. And then, of course, you have to validate in the proper way. So that was also my main assumption when I started this many years ago. So, but I very fast realized that the machine learning algorithm is not really so important. So no matter what kind of machine learning algorithm you put there in end, if you take an ECFP fingerprint, for example, there's not a huge difference if you use a random forest, or if you use a neural network or a random or support vector machines. So there's not so much to gain. So we realized very easily or very early that we have to do something on the representation, on the descriptor side. So how do we want to describe molecules, so ligands in this case? So there is always a question how and which descriptor you want to use. There's a wide bunch of descriptors. You can start with binary descriptors, which just take into account functional groups and count them, maybe like pharmacophores or toxicophores. You can go for 1D or FISCAM predictors, which just like simple things like molecular weights, polar surface area, solubility, or lipophilicity, whatever it's easy to measure. And you can take also more complicated until you maybe reach the 2D circular fingerprints, which have been so far the working house in most computational chemistry projects. So, but then I think it was 2014 or 15, I can't recall right now, I think it was 2014, when Duvna was presenting their neural graph fingerprints. This was really a breakthrough, and suddenly we thought, yeah, if you can do so much better prediction by just changing the neural descriptor by using some graph convolution neural network, then there is maybe much more to win. And therefore we focused a little bit in our first part on the developing of a new descriptor. And where do you see what we have developed? It's a little bit borrowed to the concept that has been developed by González Bombarelli. So that is learning and embedding on the ligand side, so on the chemical representations, and try to learn a sequence-to-sequence model that can represent a chemical structure in a fixed size Latent space, and from therefore you can decode into again to the chemical structure. So, but we thought, Esther never ever estimated the power, the capacity of a recurrent neural network, so they can store everything. And as Joshua Benjo sometimes already mentioned ago, they never forget. So maybe it would be better to make a more complicated task. And therefore we borrowed the concept of manual translation. So we present a chemical structure in a different form of the SMILES notation. It's a line notation that is describing the molecular graph. And by the way, can you see my mouse if I move it?
Yes, we can.
Okay, perfectly. So as depicted here on the side hand, this chemical structure here can be depicted as this Inchi key. Oops, sorry, at this Inchi key. But on the right hand side, it is now also depicted here as a canonical SMILES. So what we have developed sequence-to-sequence model basically that takes a SMILES notation in form of an arbitrary SMILES and translate it into its canonical version. So thereby we force a network to learn something and to understand what this string representations means. What is the opening bracket and so on does it mean? So it just not can memorize everything. It has to learn something about the semantic. So what we are doing, we are translating between two semantic equivalent representations of the same molecule. The nice thing is now is that can be done on a very, very large scale. So what we have done, it's completely unsupervised. We have trained this model on roughly 75 million coming from PubChem and Think. And I show you a little bit so what you can achieve with it. So on the left-hand side, on the first panel, you see the translation accuracy. If you do, for example, in the red hand, the red line shows you the translation accuracy if you translate between 2 times the same representation. So a canonical SMILE to a canonical SMILES. This is a very easy translation task, and you see it, translation accuracy are very high. But if you then, at a certain point, takes a lipophilicity as a task, for example, and takes the representation that it learns, for example, at the time point, at the time step 5000, then you see that the performance of this model, if you take this representation, is very poor in an R-square and a regression task. So, but if you then, on the other hand, take a more complicated one, so the blue one depicted, so you translate between a SMILES to a canonical SMILES, then you see that the translation is much, much harder for the network. But if you again take this representation that's learned by the translational task, then you see that the performance of the underlying predictor is much better. So, and we showed this for classification and for regression tasks, and also for a bunch more other tasks. We validated this across graph convolutional neural networks, across circular fingerprints, different kinds of clustering, not clustering, classification or regression algorithms, and also with different kinds of folding for the fingerprints. So what we received, so we received a very robust and solid fingerprint descriptor that that is very well suited for classification and regression tasks. Stuff had been published in Chemical Science, and all the stuff is also available at GitHub. Or it's also pre-trained as a fully trained model. The nice thing is that we constrained this descriptor also, that it's forced to be learned something, that it's forced to learn something of the chemical. And that can be shown very nicely. For example, if you depict the small molecule, the aspirin, that is like in the bounding box, and represents this in a chemical space, and now move along the first principal component of chemical that we have also embedded in the same space. Then you see if you move along the first direction of the chemical descriptor, then then you sample from this descriptor a new molecule, then you can generate valid new molecules. But also the descriptor is preserving something about the meaning of this molecule. So you can see here it is. In this first direction, it's a molecular size of this molecule, while the second principal component in this case is describing the polarity of the molecule. So Now, but this is now we've shown you something what we can do to describe molecules, but at the end we are interested in developing bioactivity models. So and there we'll always end up with either developing a single task model. That means you have your set of molecules, ligands, and you build for all your proteins always one model that is trying to predict, is this ligand active on this particular molecule? The problem if you do that you have to always calibrate the models across the different ones. So it's not that you can directly compare between two models. No, you have to calibrate, and you need some blood scaling or isotonic regression to make the results more comparable. And the problem is also there is that you that you cannot exploit knowledge across different measurements, across different proteins. So therefore, you could think of developing this thing now on a multitask setting. So again, your input for this model is like a ligand descriptor. Now you have for each output node, for each protein that you want to model, one output node, and there we always get this prediction is active or inactive for a certain for this set. However, if you do it like that, then you can borrow statistical strengths across the different tasks. But the problem is, what do you want to do if you want now to start and want to predict something about a new protein that was not part of your training data set? And there you cannot do anything. You have to retrain everything from the scratch, and therefore it's not very well suited for drug developing purposes. So therefore we come up with this idea, or not we. So it was basically also earlier a day by Gerard von Westen and also Andreas Bender about developing proteochemometric models. So now we always present the network a set or a tuple of a ligand and a protein, and then we can borrow strengths across different combinations of ligands and proteins that you see, and in order to predict if this protein and a ligand is active or not. That shows very nice things. But there you always run into the problem. So which kind of descriptor you want to use from the biological side, on the protein descriptor? So again, there are many, it's following the same principle again. So you know that certain proteins, if they are showing a high multiple sequence alignment score, they show lots of similarity of the amino acid in the protein sequence. So maybe it can be also that if they share a lot of similarity across the proteins, that those proteins also share some kind of active pockets where a molecule or a ligand can bind again. So in order to describe it, you can again use binary descriptors, or what people have done in the old school fashion, like taking some amino acid-based descriptor and calculate for each amino acid some kind of FISCAM properties, then take a PCA to aggregate them, and to thereby describe the sequence. So that is nice stuff. You can do that also, but we can exploit there the same principle, and that we have already done like 3 years ago or 2.5 years ago. So we took a sequence-to-sequence model and represented a protein sequence trained on 120 million protein sequences from UniProt into this fixed size Latent space. Unfortunately, we could not do this translational trick here. We only used a reconstruction trick, but it was fine. In the meantime, this concept has been exploited by other groups. George Church, they had used their UniRep embedding, or Burkhard Rost's lab with their sequence WEC embedding, and also the tape representation from transformers. Okay, but nonetheless, so now when you have it, now you can take an arbitrary protein, embed it in a fixed size and space. You take an arbitrary ligand, encode it also, and now you can combine those two embeddings and perform a regression task and see, for example, does this ligand binds to this protein or the PIC50 value of 7.7? We have presented this last year at the Learning Meaningful Representations workshop at the NeurIPS. And so the results are very extremely promising. So we benchmarked it against the Lenslink paper. And basically what you see is that the DPCM, that's what we called our model, deep proteochemical modeling model, performs very well. So we did some kind of ablation study to understand if where the strength comes. Is it the fingerprint descriptor? Is it the protein descriptor? And what you see here, that's the best if you use our unsupervised, that's called unsupervised protein descriptor, and our unsupervised compound or ligand descriptor, then you achieve the best classification performance in a random interplural splits. But it's also worth to mention that we gained the largest performance boost by using actually the compound descriptor and not the protein descriptor. And that is basically to the fact that the protein descriptor is not learning a very good hierarchical representation because it's just memorizing the protein sequence. Okay, but when you now want to apply this kind of model and on a large scale data set coming from Campbell in order to use it for repurposing projects, then you will realize that you cannot compare it across these different PIC50 values because there's a third component involved, and that is the assay in which the PIC50 was measured. This can be a biochemical assay or a cell-based assay, or what kind of tissue-based assay. So we took this large data set and included this information unencoded and then performed the regression task. And we borrowed also a little bit more additional strengths by performing on the compound embedding simultaneously some kind of an ADME regression task. So you can think of it as a multitask regression model. And the main idea behind is that you know that PIC50 binding of a ligand against the proteins is sometimes also dependent on the lipophility of a compound. And by incorporating this kind of information, you can borrow strengths about the PIC50 prediction and the ADME prediction. I will walk you a little bit through the results. So I cannot show gene symbol, but what we have basically done here, we have took our in-house data set, a little bit more than 2.2 million PIC50 measurements across a little bit more than 1,000 proteins. Then we did some kind of a compound leaf-out, compound cluster out cross-validation. And what you always see here are the aggregated R-square and Pearson correlation for one target, for the targets averaged across the different leaf-on-out folds. In order to give you a flavor, what does the Pearson correlation of 0.9 means, you can take a look on the right-hand upper panel. That is one of the most best performing targets that we have models. And a Pearson correlation of 0.6 or an R-square of 0.2, that is what you see in the lower panel. So those models are quite good in picking up the correlation in the data and also modeling the PIC50s. So therefore, we wanted to see how can you use this on holdout targets. So what would happen if we would take out one protein completely from the whole training and just try to predict it? So mimicking the scenario where you have absolutely no information about what kind of ligand binds to this protein. So, and that is shown here on this plot. So you see for some proteins it works reasonable, quite okayish, I would say. But for others, you have really run into problem. One very nice artifact that you will always observe if you work with this kind of data is the problem of censored data. You see that here in form of this kind of stripes here. Okay, now coming to the interesting topic here. So we wanted to use this kind of modeling approach in our COVID-19 activities. So it all started when we were in the beginning of February. We realized that Wuhan was closed down and we had this model already fully developed, and then we said, oh man, there is no crystal structure out there. So you cannot do docking, you cannot do like any physical based modeling. So no force field perturbations, nothing there. So we thought, okay, we can use our model at least and try it. So, and thereby we realized very early that the sequence similarity between the main proteases of the SARS-CoV-1 and 2 is very high, roughly 96% sequence similarity. So, and that was our working hypothesis that we could say, so if one compound binding on SARS-CoV-1, it might have a high chance that it might also bind to SARS-CoV-2. But additionally, we know that there is a lot of sequence similarity between human coronaviruses and also other viruses. And therefore, we systematically analyzed all data coming from Campbell that is related to the main proteases or of any viral protein. So that they gave us suddenly something like 33,000 IC50 endpoints for roughly 22,000 unique compounds coming from more than 80 viral target organisms. And here we see also the problem that they had like 3 larger assay groups, mostly cell-based and biochemical assays. And that was also our start. Okay, what we have done? We trained our model on the regression PIC50 task. For model validation, we used the leaf compound cluster-out cross-validation. And we did the same for clustering. And in order to do this compound leaf-on-out cross-validation, we clustered those compounds on ECFP fingerprint similarity into 10 different clusters. And then we did early stopping on the cluster, on the hold-out cluster set. And we trained the whole data set on 2.2 million of our internal data, plus the 30,000 external data. We oversampled a little bit the viral information because it was easy that this kind of signal get lost in this large space coming only from human proteins. So, and as mentioned before, this is a purely sequence-based approach with no X-crystal structure prediction. So on average, we received over all proteins that were models an R-square of 0.6 and a Pearson correlation of 0.8. So if we only take a look on the prediction of the SARS-CoV-1 main proteases, then the results are not that striking. So, but nonetheless, better than nothing. So the mean squared error of 0.36 and a coefficient of determination R-square of 0.3-ish. So depicted here on the right-hand side is the predicted PIC50 above the true IC50. Okay, so then we took our model that was trained and did started some PCM-based virtual screening using drug bank on approved and experimental drugs, because our working hypothesis is if we want to come up with a potential drug that has a potential treatment, shows potential treatment for COVID-19, that you have to come up and you can only use existing drugs that are out on the market because they are at least safe and do not do that harm. So just for the trade-off. So in the drug bank database, we had something like 2,700 marketed drugs and a little bit more than 10,000 experimental drugs. We predicted the bioactivity binding against the main proteases, and the results was really striking. We thought, yeah, come on, we wrap up the thing, and we just submitted, and we have one again of those publications that predict like which kind of compound is binding against COVID, as you have seen so many in the early February. But we had a little bit trick. We wanted to also come up with some kind of confidence interval of our predictions. So therefore, we have implemented some kind of a Bayesian Monte Carlo dropout, some kind of Bayesian confidence interval for the predictions. And when we applied it, then we realized that the standard deviation of our predictions is really awful. So a standard deviation of 0.7 is something that can be coming from a random distributed or uniform distributed PIC50 in a value range between 4 and 11 PCMs, PIC50s. So we realized that those models are more or less just giving random classifications. So that is it was very important for us. So therefore, we could at least test our validations. Yes, so that is already the unfortunately very not successful end of our COVID starts, but nonetheless, it was also a start of some larger activities. So based on our results, and since we already started very early on it, we were asked to join the IMI CARE project. It is a large European effort to come up with new cures for COVID, maybe not for COVID-1 or 2, but maybe for 3 and 4. So, so we are part of this very large international consortium of pharmaceutical companies, but also the top leading academic groups in Europe, where we build predictive models and also test our stuff then in large virtual screens that then afterwards will be tested. So results are, we have not any intermediate results. So far, just a summary. So at least I hope I can show you that we introduced a very nice self-supervising approach for generating chemical and biological descriptors. So at least for the chemical descriptors, they outperform the state of the art, while they have nice property. For example, they don't lead to any collisions of hashing that comes typically when you want to boil down this really large space of ECI fingerprints that are in the range of billions to a fixed size vector of 1024 or 2048. It gives you a very, very nice, easy way to calculate closeness between molecules, because now you can just calculate between these continuous representations the cosine similarity instead of having Tanimoto similarity between binary representations. I showed you that the proteochemometric model takes information in the ligand space and the protein space simultaneously into account, and thereby it can improve the predictive performance of a model. Unfortunately, in our case, we were not able to show any potential treatments using existing markers on the drugs. So none of them were predicted to bite actively. But so far, and maybe I'm completely wrong in the meantime, I think that is correct. So far, we have not seen this in the meantime. And with that, I have to really acknowledge my team. So we are a large machine learning research group, very awesome people. It's a pleasure to work with them. I really enjoy them. And if you have any questions, I'm happy to take them. But my last slide, we have, of course, several open positions in our group. So if anyone is curious, just drop me a message, please. And with that, I'm a little bit too fast.
No, great. Thank you very much for this excellent talk. There are a couple of questions that people were asking, so I will repeat them, and perhaps Oko, you could provide an answer. So the first question was, how much did it cost, and how long did it take you to train your sequence-to-sequence model on 75 million compounds?
I cannot recall any longer, because we have trained this already like 2 years ago. So I have to ask Robin, who did it that time. I really don't know, but it was not so long. I really don't know. It wasn't. But the problem is, it does not take so long any longer, because now it's pre-trained. It's on GitHub. Everyone can take it, just embed molecules, and the encoding of any ligand in this CDD space can be done in I don't know. Let me see. We did it on a billions of molecules, and it does not take that long. It's fast. So maybe it just takes a few days, a billion, but typically if you want to encode 1 million, I don't know, it should be done in less than 45 minutes or something like that.
Okay, super. Can you say anything about the hardware you are using, or is there anything special happening there?
Yeah, I can. So we have a very, very nice infrastructure. We have a couple of DGXs. But we don't use a full DGX. So the sequence-to-sequence model was trained at that time on some kind. I think it was this M100 or something like that. Or no, it was a Pascal. Like really, like 3 generations before.
Yeah, that's old.
That's quite old, but it's fast. But we have extremely good hardware.
I see. Okay, thank you for this answer. Another question is about, do you think it would be possible to improve the protein descriptor using some sort of graph representation, or do you think it is just about more sequence data to improve it?
No, forget about sequence data. So unless you don't have a very good translational task that you can use, it doesn't make sense to include more sequence data, because we already took like 120 million sequences. And yes, you can learn something, but the network is not learning the underlying representations of the protein. It does not learn. So definitely, I think having some good graphs, it would make sense.
So you think it would kind of nicely complement the information you are currently using in your model.
Yes, absolutely. So, and I think really that is a very rewarding research can be done in this field. So working on proteins with graph representations would make lots of sense for me.
Super.
I would be very curious to see new things there.
Great, thank you for this interesting question. Another question is about would be the method sensitive enough to detect the impact of a variant on drug binding?
Very nice. That, you know, that was always my idea also. Like, you know, you have very often this kind of variants in a protein that's leading to some kind of resistant mechanism. So, and I thought, yeah, we can do that. So I think this is not you cannot do that. Time being, but once we get more data on drug resistance, also having more clear meaning, what does it mean and what does it mean? So maybe you perturbate all those variants or amino acid in a protein sequence, and then you see and you measure what does it means in terms of binding, then you can probably do that. But for time being, I don't see it. And so it's just like, you can think of it like it's like one amino acid in average has a protein, a smaller protein, 500 amino acids. So it's completely lost there.
Thank you for this answer. Another question was about whether PCM models are successful in case of less amount of data. And the example is for ligands like antimicrobial peptides or antiviral peptides.
I have not, so I can tell you only like from, yes and no. It's not really clear. So I think yes, it can be very successful if you have in your large training data some kind of proteins which have a very high protein similarity to the target of interest. And then you can exploit the correlation between the target that you want to predict and the original and the trainings data. So, but if there is no overlap, then I think it's not. I would not expect there to see a bigger strength. So maybe then for this particular case, it could be even better to train a single task model that is dedicated for this particular protein.
Thank you very much. I wanted to ask you one question as well, as you were saying, like, what is your view of this kind of massive multitask trained type models versus perhaps task specific models versus you know, having a pre-trained multitask model that you fine tune? How are you guys thinking about this?
Yeah, look, for the multitask, it's always like, yeah, at the end, I think it's always becoming more complicated if you want to add something. So in principle, I like multitask a lot. So in our group, we also use multitask graph convolution and multitask for predicting ADME toxicity, ADME properties like solubility and so on. So, but at the end, it's always cumbersome to add a new task. So therefore, I for this particular task, I think I don't see a big advantage anymore in multitask. So, but it always comes at the cost that you need a good representation for the additional task that you model. So if you want to have something that all those tasks have in common, then it's, in my personal point of view, it's always better to move this kind of communality into the input.
Mm-hmm. Thank you so much.